Hey there, me again. It's Chris Gregg, and it's the last macromolecule. This is the fourth macromolecule, nucleic acids. These are the molecules that make up our genetic code. So a little bit more to talk about here. Well, maybe a little bit more to talk about here than for our other macromolecules, because this is so important and hopefully so interesting to you. Take a look at this. Okay, this diagram represents what we call the central dogma of biology. The central dogma of biology. So we've already mentioned that DNA is the molecule that's going to make up your genetic code. All of your cells have a set of DNA. They have a copy of your genetic code. The genetic code is what? All of the instructions that make you, you. They determine what you look like. They have instructions for how your body is going to go about digesting a particular molecule, carrying out a particular chemical reaction. That's all coded for in your genetic code, almost like a computer program or a recipe, if you prefer. Now, all cells are going to replicate their DNA. Anytime a cell reproduces, it has to make a copy of the DNA, right? Because all new cells need to do the same thing that the original cell did. So cells are constantly making copies of their DNA for the purposes of making new cells, which of course is a property of life. Now here's something that's a little interesting. All of your DNA, every gene, so remember we said a gene is one genetic instruction. You could say it's one recipe. Every gene codes for one protein, one molecule that does a particular job, right? That's what proteins do. They do all the work in the cell. So one protein does a job. One protein might do the job of creating a pigment that colors your eyes, or one protein might be responsible for producing, let's say, insulin, a signal for removing sugar from your blood. So every gene codes for one protein. However, what's interesting is that cells have a kind of an intermediate form of this information. So if you have a gene that codes for, let's use insulin as an example, before you actually make the insulin protein, you make another nucleic acid molecule called RNA. Okay, so you have the gene for insulin, somewhere in your genetic code, in your chromosomes, those long molecules with all of your DNA, well, somewhere there's a short segment that codes for insulin. And before making insulin, your body will make a short molecule called RNA that has the exact same genetic code as insulin. And then your body takes that RNA molecule to make the protein. Let's look at that in the context of a diagram. Now here's a cell. It's a eukaryotic cell because we have a nucleus here. Here's your DNA. We could say that's your genome. This is maybe a chromosome or something like that. And somewhere here we have a short sequence. Let's keep it with insulin as an example. We have a short sequence of this DNA that's going to code for insulin. So the first step to expressing insulin, in other words, producing an insulin protein, is to make mRNA molecules, represented here by this red ribbon, with that particular genetic code. So DNA is not going to leave the nucleus, but it produces an mRNA copy that will leave the nucleus. So you can think about it this way. If you're studying, you have your textbook. For those of you that use your textbook, you have a textbook, you have a thick textbook, okay, and you will not just copy out everything that's in this textbook, right? You're going to just make notes on specific parts of the textbook. Well, that's almost transcription. Right? You're like, okay, well, next week's test, we're going to cover carbohydrates, nucleic acids, proteins, etc. Well, you're just going to transcribe the notes that you need to study for that test. You're not just going to copy out the whole book. Well, the body does the same. It just transcribes what it needs. So if you've just had a big meal, you've eaten an entire chocolate cake to yourself, wouldn't recommend it, you're just going to make the copies of the genes that you need. Genes like insulin, genes like whatever you need to digest carbohydrates, whatever you need to digest lipids. All right, and you're going to make many mRNA copies of each of those genes. mRNA leaves the nucleus, and in the cytoplasm, as well as other compartments in your cell, there are little structures called ribosomes that we see here. Ribosomes are structures that are going to take the mRNA. mRNA will thread itself through the ribosome, and those instructions on that mRNA transcript are going to be used by the ribosome to make a polypeptide chain. You remember what a polypeptide chain is? Well, polypeptide is a chain of amino acids. So we're going to see what that process looks like next lecture. But first things first, we need to look at the structure of these molecules, just like we did for the other macromolecules. So there's two types of nucleic acids, deoxyribonucleic acids and ribonucleic acids, namely DNA and RNA. Okay, they have very similar structure, but their functions are slightly different. And of course, there's slight difference in structures that we're going to look at now. So the general structure of a nucleic acid is, here at the center, we have this pink structure. This is a sugar molecule. It's a pentose sugar. Do you remember glucose? Glucose was a hexose sugar. It had 6 carbons. A pentose sugar has 5 carbons. 1, 2, 3, 4, and 5. Okay? The nucleic acid will have a pentose sugar. It will have a nitrogenous base, okay, so a ring structure with nitrogen in it, attached at the 1' carbon, so the first carbon, and it will have a phosphate group attached to the 5' carbon. So phosphate is a phosphorus atom with 4 oxygens bound to it. A nitrogenous base is a carbon ring with at least 1 nitrogen molecule in it, 1 nitrogen atom in it. So, what are the differences between DNA and RNA? Well, it's in the name, okay? RNA, ribonucleic acid. The pentose sugar will have an OH here and an OH here. An OH on the 2' carbon and an OH on the 3' carbon. DNA is deoxyribonucleic acid. So the 2' carbon is missing the oxygen. Deoxy, it's missing the oxygen on the 2'. So that's one difference between DNA and RNA. Another difference you should keep track of is, do you remember when we said that the genetic code is made up of 4 letters? The 4 letters in DNA are A, C, G, and T. Adenine, cytosine, guanine, and thymine. Okay? In RNA, instead of thymine, they have a molecule called uracil. Okay? So, kind of 2 different pieces of information we have here. Well, first of all, you see that there's different nitrogenous bases. And they're divided into 2 categories. You have pyrimidines and purines. And I probably won't ask you too many questions about the differences, but you can see that purines here have 2 rings, okay, and pyrimidines only have 1 ring. Okay, the most important difference is between DNA and RNA, you want to see that there's a uracil instead of a thymine. If we go back to the general structure we were looking at before here, this is the nitrogenous base. This is where it varies. So you can almost think of it in a way similar to the amino acids. There was the R group, and we said there are 20 different amino acids and therefore 20 different R groups. Well, nucleotides, there are 4 different nucleotides because there are 4 different nitrogenous bases. Technically, there's 5 different nitrogenous bases, but each kind of nucleic acid has its own set of 4. All right, so DNA, ACGT, RNA, ACGU. They polymerize, so these are polymers. So proteins, carbohydrates, and nucleic acids are polymers. What does polymer mean? It means that the monomers, in this case we call the monomers nucleotides, the monomers are going to form covalent bonds with one another and make a long chain. Here's our covalent bond. We've seen it before. Goes without saying, condensation reaction. Boom, there's our water molecule. There's our covalent bond. You know that these covalent bonds have fancy names. In nucleic acids, the fancy name is a phosphodiester bond. A phosphodiester bond. What was it in carbohydrates? Carbohydrates. Glycosidic linkage, proteins, peptide bond. Nucleic acids, phosphodiester bond. And the phosphodiester bond, okay, well, it's going to make a chain just like we saw in our other molecules. Now, nucleic acids, as we go through this information, you're going to find nucleic acids, they have a direction to them. on this end of the molecule, we call that the 5' end where the phosphate is, right? And we always add, and when I say we, I'm talking about your body, your cell, we always add nucleotides to the 3' end. Here's our 3' carbon, and that is where we're going to add our next nucleotide. What did we just do there? Condensation reaction? There's water somewhere here. Okay, here's another condensation reaction. What are we doing? We're making a chain of nucleotides. This is a molecule of DNA. Actually, I made a mistake. This is a molecule of RNA, right? Because the 2' carbon has OH. You'll also notice RNA is single-stranded. So we call this the primary structure of a protein, of a nucleic acid, right? Primary structure, meaning just the linear chain of nucleic acids. Okay, so this is RNA, one chain of RNA. You see here the different nitrogenous bases. Okay, there we go. Goes from the 5' to 3' direction. Now, DNA is double-stranded. Unlike RNA, DNA is double-stranded. So what does that mean? You have your one chain of nucleotides, okay, and another chain bound to it. Okay, what's interesting about here is there's a guy named Chargaff, Erwin Chargaff, and what he discovered is that particular nucleotides only bind with other nucleotides specifically. Okay, G always binds with C, A always binds with T, CG, AT. If you want to remember this, well, you can remember my name. Chris Gregg is a teacher. If that helps you remember. Of course, you only have to remember, right, 4 letters. Not a big deal. C always binds with G. A always binds with T. This is the basis of the double strands that we see in DNA. And of course, between, what do we see here? Hydrogen bonds. Now, we know that hydrogen bonds are weaker than covalent bonds. So what happens if you heat up DNA? What happens if you put DNA in a very low pH? Well, you break the hydrogen bonds and you denature the DNA in the same way you would denature a protein. So where you would have 2 strands of DNA joined by hydrogen bonds, well, if you denature it, and your body often has to denature your DNA, it's going to pull them apart. Okay? So you can break your hydrogen bonds there. Here we see another representation of DNA, TA, GC, GC, right? Chargaff's rules still in effect. What else do you see? The secondary structure of the DNA, specifically, double-stranded, well, we say it's anti-parallel, because one strand goes in the 5' to 3' direction, and the other strand goes from the 3' to 5' direction. They go in opposite directions. Another part of the secondary structure of DNA is that it forms this double helix. And if you've seen any science fiction show that talks about DNA or CSI or whatever, they always show you that rotating double helix of DNA. So it's probably something that's kind of familiar to you at this point. You've seen the DNA double helix, and again, that is the higher structure of a nucleic acid. Okay? We said that DNA is always duplicating itself. Well, how does it do that? It takes these 2 strands and it separates them. They denature. Now, this is done with an enzyme, all right? And then we start adding nucleotides to each of those strands. And you can see that here, right? So we've just taken this double strand. We've separated it into 2 strands. And we've added nucleotides to each of these strands. And what you get at the end, well, if you take 2 strands, and you add, we call these complementary bases. In other words, A will bind with T, C will bind with G. Once you add complementary bases to those single strands, you get 2 new double strands. So we polymerize new strands using the old strands as a template, and this is how DNA is replicated. That is a really simplified explanation of what happens. Well, what happens to RNA? So we have these exposed nucleotides. Well, a lot of times RNA is just going to be single-stranded. It will be kept stable by certain molecules. So it could be brought, as we saw earlier on, it could be brought outside of the nucleus for what we call translation, the synthesis of proteins. But other times, RNA can form more complex structures with itself. So if you look here, we see a single strand of RNA, but what's it doing? It's folding in on itself. And in a way very similar to proteins that we saw, RNA that folds on itself, RNA that has this more complex structure, can have functions very similar to enzymes. Okay, so a few things to take with you. Differences between RNA and DNA. Okay, RNA is ribose, DNA is deoxyribose. 2 OHs, 1 OH. Okay, DNA is missing the 2' OH. Nitrogenous bases, so RNA uses uracil, DNA uses thymine. All the other nucleic acids are the same. DNA is usually double-stranded, and RNA is usually single-stranded. So a few little factoids there. Last thing, okay, we talked about ATP. We said when it comes to energy, your cells take a glucose molecule and they make a nucleic acid out of it. So the energy currency of the cell is a nucleic acid, and this is what's coming out of it. Look familiar? Ribose sugar, phosphate, nitrogenous base. So your body, your cells, take the energy from glucose and they add more high-energy phosphates to the nucleic acid. And this is the molecule that your cells will use directly for energy. This is called A, because of the base, T, P, triphosphate, 3 phosphates. And every time your cells need to take energy from this molecule for a chemical reaction, they'll break one of the high phosphate bonds. All right, take a look at this. But by now, you should be able to recognize the different categories of molecules, right? So take a look at this, test yourself, make sure you can recognize these molecules. Shouldn't be too hard at this point. Next lecture, we're going to look at transcription and translation. In other words, how does the body recognize the genetic code? Okay, see you next time.